Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today I am going to introduce you Chai-1, decoding the molecular interactions of life. So we will talk about Chai-1, we will discuss some results comparisons of Chai-1 with AlphaFold and other currently running programs, and I will also teach you how you can interpret the results received from Chai-1. So let's begin. First of all, this slide is about the introduction of Chai-1. Chai-1 1 actually explains the version number. So Chai-1 is introduced or released by Chai Discovery Lab, and it is AI package or AI train package. It is using AI train models, and with the help of Chai-1 you can model protein-protein interactions, protein structures, protein DNA and RNA interactions, and above all you can model protein ligand predictions or protein ligand modeling, or whatever you can also say, a kind of protein ligand docking. I'm presenting you this program, and in this series this is the third program I'm presenting to you. I have also taught you about the AlphaFold 3. I have also taught you about the bolds one, and now I'm going to teach you Chai-1, because Chai-1 claims in their results that it is producing better result than AlphaFold 3 in terms of protein and ligand pose predictions. This slide is from their paper or from their website. So the left side is ligand pose buster set for AlphaFold 3, and this is Chai-1 and Rosetta TTF fold. So Rosetta TTF fold is far behind, but Chai-1 is slightly better. If you can see, it's slightly better than AlphaFold 3 in ligand pose predictions, or predicting the best poses for a protein and ligand modeling. On the other hand, they have also tested for all atoms the Chai-1 single sequence and the Chai-1 and the AlphaFold 2.3 and AlphaFold 3. The AlphaFold 3 is tested here, and for this range or for this panel, they have tested the AlphaFold 2.3 version. I think at that time, AlphaFold 3 was not released, so this could be the one reason. But for all protein interaction, it is better than AlphaFold 2.3. For antibody protein prediction, Chai-1 is once again better, and for protein monomers predictions, it's not very significant, but slightly better than AlphaFold 2. So these were all the things. So I believe, as according to the results, Chai-1 is a bit better or slightly better than the AlphaFold 3. So you are allowed to test it. Today I'm going to teach you how can you use Chai-1, and then the last slide we will teach at the end. It's about how to interpret Chai-1 results. So let's start and first learn how we can use Chai-1 for let's say today I'm going to teach you protein ligand prediction. Okay, so let's start. So first of all, you have to open your browser and write Chai Discovery Server. I will also drop this link in the description section for direct clicking, but if you're following this video, so you can click the Chai Discovery Server, and you will have this site lab.kaidiscovery.com/dashboard. So you will see three tabs: MSA tab, template tab is still disabled, they will enable soon, and specify restraints. So if you are using MSA tab, it means you are providing multiple sequence alignment produced by you. But if you are not using the MSA tab, then right now you can use the specify restraints tab. It is by default selected. So it is using a language model, and if you are interested in some restraints, you can. Okay, so the first thing is to name a job model. Let's say I'm naming in test 1. Okay, now we are going to model a protein system with ligand. So I have opened the UniProt server, and here I will write indium 1 metallo beta lactamases. Okay, I will select this structure just by default, and here I will select the sequence, and from this window I can select the copied the sequence. Now please pay attention, you should see protein here. Now click here, paste here. So now you have pasted your sequence. Okay, now you can see copies. In case you want to predict a dimer, trimer, tetramer, etc., etc., then you can increase that number, and subsequently in chain IDs you can see A, B, C, D. It means I'm going for a tetrameric protein. If now I'm going for dimeric, but in this case I'm going for a monomer, and because it contains a signal peptide sequence, so I have to remove the first 42 residues like EG, ETG, because these are the signal peptide sequence. So I am going just to remove ETG. In your case, you have to deal your sequence by yourself. Now, I'm also interested to model uh, a ligand with it. So add molecule. Now I will open PubChem, and beta lactamases, or the indium one, contains 2 zinc ions. So I type zinc here, and in isomeric smileys you can see here. So just copy this and click here ligand and say Control V, but I'm interested in 2 copies. So it's here. Now the third thing I'm interested to include any drug, I would say. So I'm interested in penicillin. So from here you have to copy the isomeric smileys. Yes, did it. Control C, copied. Now paste it. It's a invalid character, so I will click here, and what's okay, because I have selected protein here, so that's why now it's fine. So our system is ready. So let's try Chai-1, and our system contains 1 ligand, penicillin, 2 zinc ions, and should be modeled in the active pocket in a protein sequence. Predict, and I will name it, the name of this run will be test1, test underscore 1, and predict 3D structure. Job submitted successfully. Now you can click view. Okay, this is my job, protein ligand, and it's saying it's processing, and now we can refresh this, and it may take 2 to 3 minutes, possibly. So by that time, I will download the crystal structure from the PDB for alignment or for the comparison purposes. So let me download RCSB, because we have also to check the efficiency of Chai-1 for EXS. I know. Download PDB format. Okay, so it's here. We will open it from the download folder once we receive our Chai-1 results. Still processing. So I will pause this video for a while and until this process is complete. I think it's done with. Okay, success. Now you can see this is our structure, and you can also see there are two zinc ion. Let me check. You can see here there are two zinc ions, and they are in a perfect position because they have engaged the catalytic triad of indium 1. So this is zinc 1 and this is zinc 2. And where is our ligand? So here is our ligand, you can see. Okay, now how much good is this structure? This is important. So this is the IPTM and PTM. So first of all, we are going to discuss these things, these matrices, how you will understand like your docking or your modeling is near to the real structure, or it's just a wrong modeling or a wrong docking. Okay, so from here you can see majority of the things are in white zone, and the white zone is near to 0, and this is the expected position error. So it means from Chi1, these this prediction is top class. PTM is 0.96, means accuracy is 96%. But to understand the PTM and IPTM, uh, we have to discuss this slide shortly. Okay, there is a term, its term name is TM. TM means template modeling. The, the TM score means the template modeling score given to you from the program. So what is PTM, predicted template modeling score? Remember, in our case, we received this almost 0.96. It means 96%. So what the PTM tells you, if the PTM score is above 0.5, it means that you can consider this structure. Okay? And in our case, it is 0.96. So it means that the Chi-1 recommends the highest accuracy. I will use the word highest, the highest accuracy for the structure to be used. Okay. But in our case, we have another portion that is IPTM. Why we have IPTM? Because we are using two different things. If you are modeling only a protein structure, a single protein structure, you may not receive IPTM. But if you are dealing with two different proteins, or if you are dealing with the protein and DNA or the protein and ligand, then you will receive the IPTM score. So the IPTM score actually measures the accuracy of the predicted position of the two interacting partners. It can be protein-protein complex, it can be protein-ligand complex, blah, blah, blah. The higher value is the better one, but if your value is higher than zero point 0.8, it means be confident and go ahead. If your value belongs to zero point 0.6 and zero point 0.8, it means this is a gray zone. Gray zone means that in some cases it can be wrong, but it can be right also. It's a kind of, this is what we call it a gray zone. But in our case, we got this almost 0.75 and something. Let me check again. 0.75, yes, here. So it means it is in the gray zone, but I personally recommend if it is above zero point 0.7, you can consider that gray zone towards the true positive. So you need to understand these things because only modeling is not the main goal. Like you use AlphaFold 3, or you use boards, or you use IP, this one, the Chai-1, and you thought that okay, done. No, if these scores are on the lower side, then you have to be careful for publishing or for accepting is the result as a true positive. Okay, now finally we are going to download the results. So download results, and I will receive the result here, test1.zip. So these are in the download folder. Let me generate a new directory like test_1. I will copy these two things to this directory. Now what I'm going to do, you will see. I'm going to extract. Okay, so these are the 5 predictions, and let's see. I will open PyMol, and in PyMol we will compare these things. So first of all, I'm going to open the 4DX but it contains 2 chains. So let me remove one chain, selection move chain, this, and then I have to remove atoms. Now I have only one chain. Okay, that's great. Let me make the structure, the display white. Okay, now I'm also going to, okay, rank 4 is on the top. So okay, we will select this one. Now I'm going to align both these structures, and where it's gone. Okay, here. Okay, now can wait, clipping is on, but now this one, the greenish one, is the real PDB structure, and this cyan color is the predicted one by Kai, and I cannot see major RMSD difference apart from this. But if you know about this protein, this part is considered flexible, so that is okay. It's not a major difference. Can you see both the zinc ions are placed perfectly fine, and both the penicillin and our drug, the penicillin and the L-captopril, they are also perfectly fine to me. So this was all about Chai-1. I hope you can use it. I strongly recommend. And remember, this was our third program, Boards 1, Alpha 4 3, and today I presented Akai 1. I have also an announcement that shortly I'm planning to start a course, not a course, a kind of series of protein design. Protein design is a field by which Professor David Baker got the Nobel Prize. So I need some approval from somewhere when I receive them. So you will see soon uh, A to Z tutorials of those things on my channel. So let's wait and keep watching. Share this channel if you're liking my work. I always bring the latest stuff to you, and I hope I am not disappointing you or I'm not wasting your time. Okay, thank you so much. Bye bye.